Hi, thank you everybody for being with us today. This is day 3, and today we'll be talking about highly pathogenic avian influenza challenges and opportunities. Chris, do you think you could put up my slides for the recap, please? Thank you. So yesterday we had a really great day, and we discussed lessons learned from the North American HPAI outbreak in 2014 and 2015. So the lessons learned that we discussed were by USDA, USGS, as well as Canada. We also discussed wild bird and environmental sampling and surveillance methods, ramping up our surveillance this year in light of the current HPAI circulation in other areas of the world. And then we also touched on the importance of messaging and communications, which is, I think, a good lead-in to what we'll be discussing today. And today we'll be talking about those challenges and opportunities with talks regarding environmental persistence, using genetic information to elucidate transmission, as well as discussing emerging technologies for surveillance. So, as it says here, let's get rolling. So the first talk that we'll have today is from Dave Stalneck. Dr. Stalneck is a professor at the University of Georgia and director of the Southeastern Cooperative Wildlife Disease Study. He received a bachelor's and a master's in wildlife biology from the University of Georgia and a PhD in veterinary epidemiology from Louisiana State. Dave has been involved in research on avian influenza viruses in wild birds for over 35 years and has been part of the NIH Centers for Influenza Research and Surveillance for the last 15 years. He's been an author or co-author on more than 150 peer-reviewed manuscripts related to influenza. So if we can post Dave's slides, please.
Okay. In just a second, we'll get those slides up. There we go.
There we go.
Thank you.
Thanks, Sam. And Andy Ramey, I threw this picture in just for you. That is you in the center of it. What we're going to talk about today is persistence of infectious avian influenza virus in waterfowl habitats. And this is a little different perspective than what Chelsea presented yesterday. Hers was more of a surveillance slant. Our work here has been really based on trying to understand transmission. So what we have been doing in our lab and in both our experimental infections and our field experiments is actually isolating viruses and understanding how long they remain infectious. Persistence in aquatic habitats has really two perspectives we need to think about as far as transmission within waterfowl populations. One of them is within-year transmission. Basically, by providing a connection does not require direct contact between migratory waterfowl. And my personal perspective is that this is the most important place where persistence in waterfowl environments has relevance. If you look how waterfowl kind of hop, skip, and jump down a flyway, they just basically are leaving little presents for the other ducks kind of following, and a means of transmission that doesn't require close contact. Another aspect of this, this is one that's a little more difficult to kind of get our heads around, are between-year transmission. And actually, the environment as a potential reservoir component where infectious viruses could remain infectious throughout the year and actually be waiting on breeding grounds for ducks to return. Really, the history of kind of looking at this, this is not a new concept in any way, shape, or form. It's been known for a long time that transmission of influenza occurs through a fecal-oral route through contaminated water. This was also ducks shed an enormous volume. The way viruses survive is they just flood the environment with infectious particles. And this was some work done by Rob Webster way back, trying to estimate how many infectious particles a duck would shed in a day, 10 to the 10th egg infectious doses per day in the environment. So they're just really flooding it out. This is probably an overestimate, but it's just an enormous amount of virus coming out. And also extended duration of shedding. It's been shown in greater than 28 days. More realistically, in our wild birds, it's probably 14 days for a naive duck, or 14 to 20 at best. And there's been many isolations directly from surface water reported. In most cases, ducks were present at the time, so it's kind of hard for a context here. But it is possible, we've done it in our lab, to take a mill of water from a waterfowl habitat, inject 3 eggs, and you will isolate a virus at about 1 in every 100 attempts. So what has been demonstrated in the lab about how long these viruses last? And we really kind of, this proof of concept, again, was at Rob Webster's lab at St. Jude. And I asked him specifically about this experiment, and it was done on a lark, like a lot of interesting discoveries. He basically sent one of his postdocs down to the Mississippi River from the St. Jude lab, which is about a quarter-mile walk, brought some river water back, stuck some virus in it, and then just looked for infectivity over time. And what he found is that it was viable for about 32 days in river water. We tried to refine this a little bit and try to develop some model here, and I did this for my graduate work at LSU. And basically, we have a distilled water model where we can kind of modify the chemical and physical properties of water to see what it does with flu. And these are some of the real basic kind of relationships that we see with pH, temperature, and salinity. We chose these because they're also easy to measure in the field. So if it has any application in the field, we can kind of move it that way. But what we see is some pretty consistent trends. Long story short, flu viruses remain viable for the longest period of time at a neutral pH, at the lowest temperature possible, and at 0 salinity. But there's an enormous amount of variation between individual viruses in how they respond to these variables, especially with salinity. We also have done some work with high path viruses. These are really actually early lineages of H5N1, goose guangdong. And I think I put this slide on just to show the extreme variation with viruses, with individual viruses, and how they last in the environment. And I'm just going to be honest here, we have no idea why this variation occurs within an individual virus, and it's often very, very unpredictable. And there's no relationship. High path viruses don't last longer than low path viruses, et cetera, et cetera. Okay, that's sort of the lab component, the easiest part of this. Just 3 things: salinity, pH, and temperature. So we wanted to see if what we were doing in the lab would actually work in the field. And we've done this 3 times. I did a small trial in Louisiana, and it seemed to work just fine. I mean, as far as low pH, it was predictable whether it was a field sample or a lab sample. But we had a graduate student, Seamus Keeler, and we decided to investigate this a little further. So we took it to the field in Georgia, taking water samples from waterfowl habitats throughout the state, looking at salinity, pH, conductivity, and we did this at several temperatures, and it actually holds pretty well. But it really, the predictive kind of part of this is pH. And you can see the red dots down here. Viruses in these habitats probably will not persist for any extended period of time. The RT values for these habitats and viruses, and an RT value is a reduction time of 1 log. So it's the time it takes for 1 log of virus to degrade, 1 million to go to 100,000, 100,000 to go to 10,000 infectious units, etc. So basically what we can say is, at least if you're measuring pH, some of these blackwater habitats in South Georgia, transmission is probably reduced in here because the virus is just not going to persist for any extended period of time, days instead of months. We also took this to 38 national wildlife refuges throughout North America. Seamus probably traveled more than any graduate student in history, but the same kind of relationship. So it's grossly predictable. Transmission between years, looking at a potential reservoir component. The one thing that was pretty obvious to us, we kind of concentrated on cold water habitats. And really what we're looking at is avian influenza viruses remain infected in cold water habitats for periods consisting with overwintering. Again, these are lab experiments. You can see the RT values in the bottom here, but the RT values for some of these viruses at 4C range from 178 days to 256 days. And these are all H5, low-path H5, H7 viruses. But what this is telling us that, within the course of a year, maybe only 1 log of virus will degrade under ideal conditions. But it can get too cold. And this is one thing that is pretty characteristic of viruses. You have to be careful with viruses when you have when you freeze them, and especially when you have freeze-thaws. And influenza is no exception. And this is some work that we've done just going beyond that 4C range and looking at the effect of freezing at -20 and then -20 with freeze-thaws. And what we see there is a very, very quick reduction in titer. The one point that I want to make here, and this is shown on the graph at the top, and the solid line is infectivity and how it is reduced over various freeze-thaws. The dotted line there is actually RT-PCR CT values. And what you can see there is the virus degrades, but the RNA does not. And that is why you really can't use PCR, even quantitative PCR, to look at infectivity in a water environment or environmental. You have to go to virus isolation. Okay, looking at lab work, we wanted to take this to the field and to actually see in sort of a modified quasi-field experiment, can these viruses theoretically overwinter in a waterfowl habitat? Be deposited by a duck and be infectious when those ducks are returning after the wintering period. And to do this, we did this work. Andy Ramey and I we worked together on this. This has been a fantastically interesting project. I got to go to Alaska, which is just the best part of the whole deal. To do this, we ran into a glitch, and I remember having this conversation with Andy in the lab when he was here, is that we can't put infectious virus in the field. We can't do that. So how do we get around that? We both kind of looked at each other and said, well, we can use feces. So this is what we did. So basically, what we did is we took natural lake water in 2 sites. We've been working in Minnesota at Agassiz National Wildlife and Eisenbeck National Wildlife in Alaska. We filtered that. This does not contain any of the biological components, so it is that artificial. That part of it is artificial, but we filtered this, We inoculate it with feces excreted from a duck. So what we're dealing with is the virus that actually comes out of the duck cloaca. This is as natural as it gets. And we actually diluted this in 40 mils. This is a swab diluted in 40 mils of filtered water. We aliquoted these in smaller tubes, and we bagged them up and sunk them in lakes in both Alaska and Minnesota. We've done 3 trials, again, in these things, and we actually retrieved replicates over time. And where we concentrated on is this sweet spot. And we did this based on the lab work where we saw the greatest longevity of infectivity at 4C. And so what we did is we actually put these in metal cans and sunk them in the lake in approximately 2 to 3 meters of water, and tried to get to the point where it's shallow enough that a duck may actually have contact with this thing if it returns, but deep enough where we would not get the water would the cold water would be maintained, but it wouldn't go through a lot of freeze thaws. So basically, we retrieved these aliquots throughout time and did virus isolation on them. And the long story short is that avian influenzas can maintain infectivity through the overwintering period. And this is not only determined by isolation and egg inoculation, but we actually took the virus and with the Southeast, through collaboration with the Southeast Poultry Lab in Athens, Georgia, took some of that water, gave it to a mallard duck, and infected the mallard. It's interesting to just put this in a little perspective. We did some titrations on these fecal samples, these diluted fecal samples that we put it in here. And the highest titer that we saw entering this water was 3 logs of virus. So we're dealing with really small volumes of virus, and they can go over the year. Not all of them. It's probably about 5 or 10% of them make it. So basically, AIVs can theoretically maintain infectivity for a greater 1 year, fall migration to fall migration. Okay, I just pretty much showed the most simplistic look at all of this. But I really want to just concentrate now and end up on what we do not know and what makes understanding the environmental component so complicated. And Chelsea kind of mentioned this, that these are really complex ecosystems. And trying to figure out where to sample and where things may lie is a tough thing to do. So the questions I want to just go through really briefly are there specific ecosystem components that are important in the transmission cycle? What are the potential effects of viral aggregation related to detection and transmission? And this topic has come up before, and it's going to be fun to kind of delve into it a little bit. Would reducing overall transmission of low pathogenic viruses by environmental manipulation enable transmission of highly pathogenic avian influenza transmission? I'm going to look at population immunity here. So are there specific ecosystem components that are important in the transmission cycle? Long story short, we don't know. With sediments, lots of RNA detections, but few reported isolations. It has been isolated from sediments, and it's been isolated from pretty strange sediments and pretty strange substrates. This is some work that Peggy Paulson did at Delaware Bay, where we have influenza in shorebirds, where we actually took sand cores and tried to isolate influenza from 1 centimeter below the surface. And you can do it. And what's interesting about that is you can actually isolate it from sand cores that have gone through tidal flows, that have been flooded several times. So it's maintained in that environment. Aquatic vegetation has received hardly any emphasis at all, including things that ducks eat. And these viruses do stick to things. And a good example of that are feathers. If you pick up feathers from, say, a banding site, they are covered with virus. I mean, they often you can isolate 2 or 3 viruses from a single feather. I don't think anyone has looked at smart weeds or aquatic vegetation that ducks eat, but it probably would be worth looking at. And Chelsea mentioned this a little bit about all the things in water, but bacteria, the effects of bacteria on influenza infectivity, algae, biofilms have not been investigated at all. And this is probably a really important thing. It's probably going to be kind of eye-opening when we move from more of these experimental controlled studies to really raw water that have all these components associated with them. And a good example, these graphs over here, the blue line represents distilled water. The green represent sort of field water that's filtered. What that red water represents is a pond. Just take the water out, put virus in, and run these same experiments. And you see that these viruses degrade a lot faster. Now, the person who did this, Seamus Keeler, was a smart graduate student, and he picked the nastiest pond I could think of to pull this water out of. So there's probably going to be a lot of variation on this. This is probably an extreme case. Biofilms, viruses get picked up in biofilms. They may become more environmentally tolerant in that. We don't know. And then there's all sorts of other biological components, insects, filter feeders, etc. And there's been very limited and conflicting results in this, and ranging from bioaccumulation to total inactivation. And an example of this is some work with corbicula that we did in the lab here. With this clam pretty much draws influenza out of the water in short order and inactivates all of it. I mean, completely inactivates it. And we've done this through basically virus isolation and feeding trials with susceptible ducks. Contrary to this, zebra mussels seem to bioaccumulate a little bit. So there's probably a lot of work here. As a management tool, I would not recommend introducing either of these to your ecosystems. I just want to touch upon this because this is an area that hasn't been even looked at as far as environmental contamination of influenza, and that are viral aggregations. And long story short, viruses may not remain as single viruses. They actually may glom onto each other and attach the thing and really become complexes. And this can potentially affect the quantification of viruses, the way we measure them. If we have 100 infectious particles suspended in a liquid, we'll measure it as 100. If they're all together, we're going to measure that as one infectious unit. So it can really throw things off as far as detectability and measuring these things. It's been reported that this type of event will also increase virus survival. It could influence the distribution in the environment, whether it's attached to inorganic particles or whatever. And some interesting things I just kind of threw in. It can also affect reassortment because an H3 virus and an H4 virus may be present in the same aggregate and be taken in by a duck in the same infectious unit. And this may be why we get so much reassortment in ducks. And then there's something called complement reactivation, which has been shown in the lab with flu, but not in the field, where actually if you have a complete genome, but a non-infectious particle, and you have an infectious particle, and both are taken up in a virus, that genome, the genome of the non-infected virus can be incorporated and can be basically rescued in the deal. Okay, would overall transmission of low pathogenic viruses by environmental manipulation enable transmission of a highly pathogenic avian influenza virus? And I really, again, want to talk about this idea of population immunity here. And because it really affects not only how much virus is put into it, that a duck excretes into the environment, but how much it might take to infect a duck. And this has been shown by us and a lot of other people. And I kind of put this in this figure in the bottom as sort of a timeline to kind of go over, kind of orient you to these experiments. what we start with in our model is an H3N8, and we do this for a reason. On a timeline in a normal year, H3N8 is the predominant virus. H3N8 and H4N6 are the predominant viruses present in waterfowl populations. This is if you're basically sampling in August and September. This is what a duck is going to be exposed to. And this is where infection prevalence is highest. So this is what they're primed with. So what this does, if you infect a duck with H3N8 and then challenge it with an H4N6, not only will it take more virus to infect that duck with H4N6, but if infected, a duck will actually excrete a lot less virus. So it can potentially affect transmission. If you hit it with the same virus, you don't get any replication. And this is an interesting one. If you hit it with an H4N8, you not only get an effect probably related to the antibodies or the immune response to the H4, but also neuraminidase. And with this matched neuraminidase, you will get no replication whatsoever. You see this with H1N1. This is some work that was done in Canada. Definitely actually reduced the infectivity and the transmission of H5N1. And what's really interesting, if you do multiple exposures across time, H3, H4, H10, and 7, and this is not something that's not occurring in nature, and challenge them with the H5N8, we got no replication whatsoever. So the reason I bring this up is that natural exposure to low pathogenic avian influenza may actually be a very important prevention tool. And when we talk about management, as we especially talk about environmental manipulation, we can't look at these systems just based on one virus. What's H5N8 doing? What's H5N8 doing? Because all these other ones will probably affect the outcome. So it's a complicated system that we really don't understand. I don't think anyone is smart enough to recommend something when you take it all in perspective. So can we manipulate waterfowl habitats to effectively reduce transmission of a highly pathogenic avian influenza virus? Well, I think we need to look at this maybe again on two levels. Does day-to-day waterfowl management in North America affect avian influenza transmission? Probably. By increasing density, we probably do increase transmission. If we're increasing transmission of H3N8, it may not be a bad thing. The other thing is, it's funny when we work a lot with banding operations. As a wildlife biologist, when I see 300 ducks caught under a rocket net, I say, that is fantastic. Very cool. We got a lot of samples. That was really neat. But as an epidemiologist, I look at it and go, my God, that is a fantastic transmission machine that we just built. So I think we need to kind of keep these perspectives in the back of our mind when we're working in the field. And I think at present it would be very difficult to define or support any large-scale habitat manipulation to reduce HPI, H5 transmission, or environmental maintenance. And very, very difficult to evaluate effectiveness. There are just too many unknowns. And with that, I'm going to quit. And I think do I have time for one more statement here? I want to just relate something that was said yesterday regarding funding. We have been extremely lucky in getting competitive results from USDA, Centers for Disease Control, and NIH for sustained influenza research and surveillance. And I cannot emphasize how important that is. It was brought up in the conversation yesterday. One other point I want to make is regarding the NIH Centers of Excellence. We have been actively involved in influenza surveillance in North America forever. And through the new NIH centers, there is not going to be anything left. And what is leaving our surveillance, our surveillance toolbox, are 40 years consecutive studies of influenza in ducks in Canada, shorebirds at Delaware Bay, 20 years of repeated surveillance in ducks in coastal areas of Louisiana and Texas and in Minnesota, and probably 40 years of continuous influenza surveillance in the Mississippi Flyway by Dick Slemons in Ohio State. So there's going to be, outside of what USDA and what USGS is doing, there's going to be also a big void of work coming from NIH. And that's all I have, Sam. Thank you.
Thank you, Dave. That was an excellent talk. Unfortunately, we're running a little bit late, so we'll have to leave questions until the end. And I'd like to move on next to Dong-Hun Lee. He's an assistant professor at the Department of Pathobiology at the University of Connecticut. He was a postdoctoral fellow with the Exotic and Emerging Avian Viral Diseases Unit at U.S. National Poultry Research Center in Athens, Georgia. So all good things come through Athens, apparently. So his current research interests are the infectious disease ecology and epidemiology of viral and bacterial pathogens in animals. So please take it away, Dr. Lee.
Yeah, thank you for giving me this opportunity today. My name is Dong-Hun Lee. The topic of my presentation today is using genetic information from wild bird surveillance to understand transmission of AI viruses between wild birds and poultry. So domestic birds, such as chickens and turkeys, could become infected with avian influenza viruses through direct contact with infected wild birds or other infected poultry, or through indirect contact, for example, contaminated feed or clothing and equipment that have been contaminated with the viruses. Then how can we track the spread of viruses? Using viral genome sequences and various phylogenetic analysis, we can trace the origin and evolutionary history and spread patterns of influenza and any other pathogens. Examining variations, genetic variations across genomes supports surveillance studies for better understanding of mechanisms that lead to introduction and spread of viruses. To do this, we should merge the genomic data with epidemiological data to determine how the virus is spreading. And this approach also enables us to find out what the emerging strains are, where they have come from, and how they have mutated to be more virulent at genetic level. So the basis of any genomic surveillance approach is the technology needed to detect genetic changes of target pathogens. The increased availability of next-generation sequencing allows rapid and economic genome sequencing of influenza and other viruses. And the high throughput of NGS, next-gen sequencing, has not only the capacity to produce an enormous amount of sequence data at much lower cost, but also to comprehensively detect all classes of genomic mutations. Once the raw viral genome sequences are obtained from next-gen sequencing machines, we need to use computer programs to assemble the genome sequences and identify genomic mutations. Again, phylogenetic analysis can be used to assess the origins of a set of pathogens, patterns of geographical spread, and host species jumps, and evolutionary history. And many of the computational tools and statistical inference methods developed under phylogenetics and molecular evolution are routinely used to show evidence for epidemiologic relationships of the casualties of infectious disease. In evolutionary phylogenetics, changes in nucleic acid sequences observed over time in viruses are assumed to be inherited events, transmission. For example, let's say the first one is the index virus of avian influenza outbreak. And as time passed and virus spread, mutations accumulate in the genomes of viruses. The first one, the second line has 3 mutations compared to the index virus, and one more, and then one more, so 3, 4, 5 mutations. And then let's say these viruses were identified from Alaska, Mexico, New York City, and Canada. And these genetic sequences suggest that wild bird origin virus most likely spread from Alaska through Mexico and then New York and then Canada. On a smaller scale, let's say these viruses were identified from wild birds and poultry farms, Farm A, Farm B, and Farm C. And these genetic sequences suggest that wild bird origin virus was most likely introduced to Farm A first, and then farm-to-farm transmissions occurred from Farm A to B and then to C. So genome sequences can play a critical role in our understanding of viral evolution and spread. Here's another example. Let's say a new avian influenza virus strain emerged and detected in these birds in red, and then spread to orange, to yellow, to green, and then to purple. As time passed and spread, mutations will be accumulated in the genomes of viruses. And we can build a phylogenetic tree based on this information. And track the spread of viruses. But genomic sequence data are limited utility without accompanying metadata. So metadata include both information about the process used to derive the sequence, such as laboratory and data analytic tools used, and information about the sample, data of collection and sample location, for example, and which is particularly important for epidemiologic investigation. And metadata can also include host information, such as species, age, sex, and migration history, and along with clinical information, such as types of symptoms, low pass versus high pass, and history of vaccination if it is poultry. If we can add more information in the tree, metadata, such as time and location of each virus, as you can see here, we can quantify and understand the spatiotemporal dynamics of virus spread. So the recent application of genomics in epidemiological investigation of 2014-2015 high-pass outbreak in the United States highlighted its usefulness. So this map shows our high-pass AI detections in the US during 2014 and 15. But I'll skip this slide since we have already talked about it yesterday. So a total of 3 new rheosurtans were found in North America, produced by genetic reassortment between East Asian high-pass clade 2344 H5N8, and North American low-pass AI viruses. The first one, in the box, this genotype of H5N8 virus was identical to East Asian H5N8 virus, but we found this novel reassortant H5N8, 5N1, and H5N2, which were mixed with wild bird origin North American viruses. The reassortant H5N2 high-pass AI viruses predominated during the 2014 and 15 outbreak. So phylogenetic analysis and understanding of waterfowl migration patterns suggest that the Eurasian H5N8 virus emerged in late 2013 in China, spread in early 2014 to South Korea and Japan, and reached Siberia and Beringia by summer 2014 via migratory birds. And 3 genetically distinct subgroups emerged during breeding season and subsequently spread along different flyways during fall migration 2014 into Europe, North America, and East Asia, respectively. Then we used a molecular epidemiological approach involving genome sequences and outbreak information to determine the evolution and spread patterns of viruses in the US. NVSL USDA sequenced complete genome of 268 viruses by next-gen sequencing, and we used a Bayesian phylogenetic model to estimate viral transmission between wild and domestic population and assess the most likely route of spread. In this model, we defined geographic region and host type as discrete categories and estimated viral migration between these categories, including Pacific chicken, Pacific turkey, Pacific wild bird, backyard, and game bird, and Midwest chicken, Midwest turkey, and Midwest wild bird. We reconstructed this phylogenetic tree and summarized the viral migration in this heat map. Each column represents 1 month, December to June 2015, and with rows representing the viral migrations between groups. You can see here the color codes. For example, this is a purple to blue, means Pacific wild bird, to Pacific turkey, at the bottom. And in this heat map, yellow color indicates more viral migrations. So in December 2014 and January 2015, there was an exchange of viruses between Pacific wild birds and Pacific chicken. And then virus most likely migrated from Pacific wild bird to Midwest turkey during January to March 2015. And then virus most likely migrated from Pacific wild bird. After viral migration from Pacific wild bird to Midwest turkey during January to March. And then there was extensive virus exchange between poultry farms in the Midwest. So we visualized the genetic relatedness of viruses using this phylogenetic network analysis based on flanked genome sequences. Yellow is wild bird isolate, and purple is backyard, and green is raptor, and red is poultry. Okay, and the index H5N2 is at the left. So this is a Northern Pintail Washington H5N2 virus found on December 15, 2014. So during the late 2014 and early 2015, H5N2 viruses circulated in wild birds, spreading directly into backyard and commercial poultry farms. And then it disseminated to Midwestern states and triggered a large-scale farm-to-farm spread. As you can see, lots of red circles are directly connected to each other. So our analysis of virus sequences and epidemiological investigations show that long-distance migratory birds played a major role in the global spread of avian influenza viruses in 2014, including from East Asia to Pacific Flyway. And after introduction to Pacific Flyway, multiple introductions of high-pass AI H5N2 into Midwest states occurred during March to June 2015, including transmission to Midwest poultry farms from wild birds. And once established in poultry, the virus rapidly spread between turkey and chicken farms in neighboring states. Okay, now let's move on to recent H7 high-pass AI outbreaks in the US. So in the United States, 3 North American lineage H7 viruses caused recent high-pass AI outbreaks, H7N8 in Indiana in 2016, H7N9 in Tennessee and Alabama in 2017, H7N3 in South Carolina in 2020. So during each of these outbreaks, a single event of mutation to high-pass AI virus was documented with 414,000 into 16 and 253,000 into 17 and 337,000 into 2020. Birds being depopulated as part of outbreak response or succumbing to the virus. The full-length genome sequencing and phylogenetic analysis were used to trace the origin of each H7 outbreak. So phylogenetic tree here indicate that all these viruses most likely originated from North American wild bird AI viruses and genetically distinct to each other. So which means each outbreak was caused by independent introduction of the virus from wild bird to poultry and evolved into high-pass viruses. So the continued active surveillance of AI virus in wild birds and poultry, combined with timely generation and sharing of complete viral genome sequences, are crucial. And these efforts have led to the detection of new virus introductions and the emergence of novel real-world viruses. So enhanced active surveillance and rapid molecular characterization of viruses would be required to monitor the spread and evolution of high-pass AI viruses and to inform the design of improved prevention and control strategies. Thank you for your attention. This is all I have for today.
Thank you so much. We really appreciate that talk. In an effort to keep us on time, let's take one question and then we'll take our break and we'll make it a 5-minute break. Do we have any questions either in the chat or on the audio?
And as a reminder, if you would like to ask a verbal question, you can do so by using the WebEx raise hand icon.
There is one question in the chat, and that question is, any human cases documented to date?
As far as I know, there's no human case of clade 2344 in the United States, and based on CDC's papers, it's not replicating in ferret models too. So, yeah.
Okay, thank you very much. Let us reconvene at noon, so that's 4 whole minutes, and we'll get back to it. We've got some more great talks on the way, so if you can go take a bio break and get any caffeine you might need and come back at noon Eastern. Thank you. Hi, everybody. Thanks for joining us again. I hope you had a brief little break. Next up, we're going to hear from Dan Walsh. He's a quantitative ecologist with the U.S. Geological Survey's National Wildlife Health Center. And he received his undergraduate degree in fisheries and wildlife from Michigan State, master's degree in fishery and wildlife biology from Colorado State, and a master's degree in statistics and a PhD in fisheries and wildlife from Michigan State. His research focuses on developing and applying novel statistical approaches to inform wildlife health management activities. And Dan's on a bit of a speaking tour today. We've already heard from him once today. So welcome back, and take it from here.
All right, thanks, Sam. So I appreciate the opportunity to come and talk about some of our work that we've been doing around enhancing AI surveillance and risk reduction. And I was asked to talk about emerging technologies around those topics. And so I'm going to focus on two different kind of studies, one in terms of some novel statistical or machine learning methods, and then the other in terms of some remote sensing and big data techniques. So one of the primary surveillance goals oftentimes we think about avian influenza surveillance is identifying new or novel emerging AIVs. And obviously this is owing to the importance of these viruses in terms of human, domestic, and wild animal health. And it's well known, as we've heard from the earlier talks today, that water birds are a natural reservoir for these AIVs, and their behaviors of intermixing on breeding grounds and these large migratory movements make them a perfect vessel for reassortment of the viruses as well as global spread. And so a lot of surveillance programs, both passive and active, target these species with the idea that if we have an early warning of these emerging AIVs from these species, we can hopefully develop suitable countermeasurements to protect the health of domestic animals and livestock. However, these national surveillance programs are costly and difficult to sustain. So anything we can do to help improve the efficiencies of these programs is an important consideration. So just a brief overview of how we actually determine or identify novel viruses, and I'm not a virologist, so I'm just going to summarize this as I understand it as a statistician, is that currently what we do, we go out in the field, we sample birds, collect cloacal or oropharyngeal samples, we bring them back into the lab, and then we extract the RNA. At that point, we test for the presence of this matrix gene using RT-PCR techniques. And anything with a CT value less than 35, we consider that a cycle threshold value less than 35, we consider that to be a positive sample. And we test for whether or not H5 or H7 subtypes are present, once again via PCR. Any sample that is less than 35, we inoculate into embryonating eggs and then try to recover the virus from the allotonic fluid of the egg using hemagglutination assays, which is then confirmed once again by matrix PCR. So this is an involved process. It's time-consuming, expensive, and it's difficult. So if there are things that we can do to improve our efficiencies around this process, there'd be great benefit associated with that. So machine learning or statistical learning are methods that we use to detect patterns in data. So we're effectively trying to model systems and, most importantly, produce accurate predictions of the outcomes arising from these stochastic processes of the system. So our thinking was, can we use these methods to enhance the efficiency of active surveillance, and can we accurately predict the likelihood, specifically, that a sample that comes into the lab will yield, via a virus isolation, the avian influenza virus. And what that would do then is allow diagnostic laboratories to be more efficient and focus their virus isolation efforts on the samples most likely to yield results. So what we did was we took data that had been collected historically from 2006 to 2007 through active surveillance. Many of the folks on the line were involved with that and provided samples for that, which we're highly grateful, because it provided about 25,000 individual bird samples to train our model. And the features that we used to provide information and help us predict whether or not we could isolate a virus included things like sex of the bird that was sampled, its age, the type of bird, geographic location the sample was collected, and then the cycle threshold or CT values generated by the real-time PCR used for the matrix gene. And the virologist, Han Ip, also included samples in testing for virus isolation from this pool that exceeded the 35 threshold that's the standard Nalm protocol. So that allowed us to evaluate what's the impact of this threshold on our ability to isolate viruses as well. So the response variable then, as I've said, is essentially whether or not we can successfully isolate a virus from these cloacal and oral pharyngeal swabs. So the method that we ended up settling on was what's called a gradient-boosted tree, and we looked at a wide range. Machine learning is a rapidly expanding field of artificial intelligence, but we looked at a wide range of different techniques from very simple logistic regression all the way up to some complex neural nets. And this technique was the one that yielded the best predictive power. And what it is in kind of layman's terms, it's just we use a sequence of these classification trees to produce a prediction. And the principle is that we can combine a bunch of these kind of weak learners, weak classifiers, to form a committee of learning algorithms that produce better results than any single classifier. And the handy thing is that we didn't have to write our own code. There are packages available in both Python and R and other languages as well to do this. And so we use the XGBoost package from program R to train our data. Now, with all these machine learning algorithms, there's all kinds of different kind of tuning parameters, knobs and dials that you can adjust on the algorithm to improve your predictive power. And so we used a cross-validation approach where we leave out 10% of the data, train on 90%, and then change the tuning parameters. We did this 100 times to allow us to determine what's the optimal value for those different parameters. And the results were actually a pretty good predictive model. Our out-of-sample predictive accuracy, so that's our ability to predict that 10% of the sample that the model never saw, that we didn't train on, was about 93%. And that's the area under the curve. And then the end sample, this is when we use all the data, so the model saw all the data, and we just predicted the results for the data that the model was trained on. Of course, we'd have a higher prediction power, and that was 96%. But mainly, maybe the more interesting thing is that when you look at the features, those characteristics of the samples that we used to model, we were able to identify that CT value in the MA-PCR was the most influential feature in terms of predictive power, and that's what's shown here on the right in the graph. And then the longitude and latitude also had some predictive power, but it's much more limited compared to that MA-PCR result. And then the rest of the features relatively did not contribute much in terms of our ability to predict the likelihood of isolating a virus. So here's kind of, if we look at just the MA-PCR results and how, as the CT value varies, our probability of virus isolation varies, you can see that for a value less than 20, we tend to have a shoulder on that curve, and then as it gets greater than 20, we see this decline in the likelihood of virus isolation. One of the interesting aspects of this too was kind of the ability to look at samples that were above the standard Nalm 35 CT value cutoff, and that's what I'm showing here. Is that at a value of 35, based on our model and our data, there's about a 16% probability of isolating the virus if that matrix gene TCR yielded a CT value of 35 or higher. And then you can see as you go down, it obviously decreases. And here's kind of the impact of geographic location on the probability of isolation. And you can see the light colors are higher probability of isolation of the virus, dark colors lesser probability. And so there's kind of the central U.S. tends to be higher, and then southeastern Alaska. So what are the implications of what I've shown in these modeling results? Well, first of all, it's kind of neat that we can look at these basic sample attributes and use those to predict the likelihood of our virus isolation in the lab. And it's very clear from our results that this MA-PCR CT value is a very powerful predictor. But location or time, and I should say location and time here are confounded because these are migratory species, and so they're sampled during migration. So the location time is also important. But it validates, too, that the screening by the RT-PCR for the matrix gene is a useful screening test, though we may be missing some viruses just by the way that we've set our cutoff values. And that could be good or bad. There's trade-offs in that. I'd also, geographic location has some influence, not a large amount compared to the MAPCR value, but if you're going to apply this, into a much more focused region, you probably want to train on your local data to improve your predictive power locally. But the kind of the big take-home message is, we can use these machine learning techniques to help us prioritize samples, and that's particularly important when resources are limited or lab capacity is limited. So I guess my big take-home message is that we can use these statistical methods, these AI machine learning methods, to help us enhance our efforts around AI surveillance and hopefully help sustain them through time. And so here's the reference for the paper. It was published a couple years ago in Transboundary and Emerging Diseases. So I want to switch gears now and just kind of finish up with a little bit of a different study. And this is one that's ongoing, so I don't have a lot of results to report, but I think it fits well into this kind of emerging technologies bin that I was asked to talk about. And this is a project that's a collaboration with Cornell University and USDA, Vet Service and Wildlife Services. A lot of those collaborators are actually on the phone today. But the idea that we're trying to do here, and what this project is intended, is to see if we can create near real-time, essentially personalized risk assessments for avian influenza that's posed by migratory waterfowl migration, and then essentially put that information in the hands of poultry producers. The idea that they can tailor then the biosecurity efforts to the commensurate risk associated with waterfowl migration. And so this is based on the principle that if we're going to actually prevent transboundary disease and cross-border or cross-boundary transmission between domestic and wildlife, the power to prevent that lies at the hands of the producers in their individual facilities. And we provide them information now that says, there's a high risk in the fall, high risk in the spring, but having long-term sustained biosecurity is oftentimes not economically feasible. And you also run into things like biosecurity fatigue. So our idea was, can we actually protect the food supply without vilifying birds, wild birds, by providing this kind of near real-time risk assessment, integrating two large data sets that I'll talk about in a minute. And then we also see some ancillary benefits from something like this, in the sense that we could provide these risk assessments also to the biologists conducting surveillance on the ground to say, here's where we're predicting birds are and will be, and so these are the areas to target your surveillance efforts, and therefore increase efficiencies along those lines. And so the first big data stream, kind of this remotely sensed data stream that we're going to leverage is NexRad radar data. So these are weather satellite or weather radar stations. There's 143 of them spread out around the continental U.S. And our collaborators at Cornell have already developed the kind of backbone to do this. They have some great algorithms developed to essentially distinguish nocturnal migrant bird species from all the other things that could be in radar signals, insects, precipitation, etc. And so that work's already there. And so that obviously provides a way, if you think about like the information you receive on weather, for example, you can go on your phone, you look at your app, and you say, okay, here's my location. Oh, look, there's a storm coming. The same kind of principles that we're proposing here, except instead of having precipitation or snow or whatever it may be, we're talking about migratory bird movement. The challenge, though, with just serving this up as it is that this is all migrants. These are all migratory birds. And so the next step is saying, how do we distinguish the waterfowl, those high-risk species in terms of AI, from all the other signatures of birds? And that's where the second kind of big data set will come into play, and that's eBird data. So this is a citizen science database that was created to document bird distribution, abundance, and habitat use, as well as trends, population trends. And there's millions of observations entered globally. And so this information provides that critical species information that we're lacking from the radar data. And so our efforts are now to look at how do we merge these information streams to get what we want, and that's the predictive power of waterfowl migrations. So here's an example of some eBird data just to show you what it looks like. I showed you the radar data. Here's the eBird data. These are snow goose reports in early March, kind of the locations of them in the lower 48. And here is a kind of a time series from mid-March to mid-April, and you can kind of see those isoclines. The lines are kind of isoclines of migration as the snow geese migrate north. And so you can see how combining this with that kind of core spatial but high temporal resolution of radar data will give us we're hoping will give us a powerful tool for providing these producers with near real-time risk assessments about the location of migratory waterfall and the risk that poses to their individual facilities. Obviously, one of the key parts of this bill is how do we serve this, and so we've been working on the application, the web application, to do that. Here's a couple examples of some wireframes that we've been playing around with, kind of building on this lights out framework for those that are birders. They know that there's this lights out app where you can, if migratory birds are passing through, they encourage you to turn your lights out to avoid collisions with buildings and whatnot. So we can use the same framework. Now we just make that, instead of a risk of a collision with buildings, we make an avian influenza risk. And we have now in a subscription service where you can get alerts. This is for lights out, but we're going to do the same thing for AI, where if you're interested, the producer or biologist could subscribe and say, okay, I want to alert when migration intensity is high at this location. And so you'll get the alert on your phone. So there's lots of efforts ongoing around kind of how this should look in user experience. So that's kind of where I'm going to, I guess, finish up here. I think there's lots of opportunities for these emerging, whether it be statistical or machine learning models, as well as a lot of these remotely sensed data sets, to really help us enhance how we think and about AI surveillance, how we manage it and try to protect both our wild bird, the conservation of wild birds, but also our food supply and the livelihoods of domestic producers. So with that, I'm willing to take any questions.
Thank you, Dan. We have time for 2 questions. Do we have any questions out there? I think you've wowed everyone, Dan. Okay. Well, you get off scot-free then.
All righty. Thank you for your time.
Great talk. All right. Up next, we have Diann Prosser, and she's a research wildlife ecologist at USGS Eastern Ecological Science Center in Laurel, Maryland. Some of us used to know that as Pathogen. Diann's research focuses on integrating field studies and spatial modeling to answer questions related to wildlife stressors such as climate change and disease. I will hand it over to you now. Thanks, Diann.
Great. Thank you, Sam. Thank you, everybody, for hanging in there. This is the last presentation of this series. And there have been some amazing talks, really informative over the past 3 days. I'd like to thank the organizers for inviting me to contribute some of our work here. Quick logistical piece. My family's on leave, and I'm in a remote location with one cell bar and really poor internet connection, so my fingers and toes are crossed that I am going to stay with you guys. Otherwise, Chris will have to take over moving my slides. So today I'd like to share with you some of our work that investigates wild bird movements and avian influenza viruses, particularly focused on relationships at the wild to domestic bird interface. And here specifically, I like to focus on this interface, right, because it's a port of entry for viruses, avian influenza viruses, to enter the agricultural system, and depending where you are globally and what viruses you're talking about, maybe also to the public health sector. And now with the evolution of these H5 clades, we have the potential for spillback back to wild birds. Aha. And hang on. Okay, there we go. So my work here started in the mid-2000s when the H5N1 virus was starting to ramp up. So a quick history here, looking at this OIE map in the top left corner. In 1996, the virus emerged in domestic geese in southern China. And things were quiet for a little bit until in 1997 in Hong Kong, there were events in poultry, but also 18 human infections, whereby 6 of those individuals ended up passing away. So Hong Kong stamped out pretty quickly the virus, and things were quiet until around late 2003 or so to 2004. And the virus was detected in 6 Asian countries, China and some of the surrounding ones. And in 2005, there was the first large outbreak of H5N1 in wild birds up in central China at Qinghai Lake. So this was the first large-scale event since that one that Tom DeLiberto mentioned that happened in turns unrelated to this virus back in the 1960s. So because this happened in wild birds, and then later in that fall, virus spread, H5N1 virus spread out of China, out of Asia, up through Mongolia, through Russia, and then hit Europe towards the end of the fall and into the winter, eventually moving then on in 2006 to Africa. And then the virus circulated in this entire region for a number of years until late 2014, when the descendants then and some mixing entered North America. And I won't go beyond that, as there were some great updates on what's happening now. But so that's kind of where I started, is when this global debate on wild birds emerged. They were implicated in the movement of the virus, yet at that time there really was a lack of information on wild bird ecology as far as their movements in these different regions. What might the pathogenicity of the virus be in different species? And the virus was detected in a large number of wild bird species in Asia at that time. If they could survive the virus, could they move with it? If so, how far? What were their migratory connections? And what was the relationship at the wild to domestic interface? So really at that point, we had to go to where the virus was to study some of these questions. And so my first study site was there at Qinghai Lake, that large event. And so this is a great map on the left here. It's Google Earth map with gridded livestock of the world overlaid on it. So the yellow to brown shadings show you poultry densities on a large scale. And you can see those blank areas, the Qinghai Tibetan Plateau, the western part of China, Mongolia, very low human populations, and very few poultry on the landscape. And that's where Qinghai Lake is located. That bar-headed goose colony where more than 3,000 geese died is in a landmark refuge there, and also Wetlands International site. And so, of course, we wanted to investigate movements of wild birds from that region, but also if you see that blue circle down to the right in the very brown, poultry-dense, rich area. That's the lowlands of southeastern China, where there's lots of rice paddy agriculture and integrated free-range duck farming. Also, people live very closely on the landscape with their agriculture. And it's a very important wintering site, especially for migratory birds. In fact, the majority of endangered Siberian cranes winter in this area. So we had 2 different study locations. We wanted to understand the movement of wild birds from these areas where there were H5N1 outbreaks. And so we marked about 125 waterfowl from these regions. And then moving forward, that bottom map on my slide with a whole bunch of different colored lines, that was where we had partnered with the United Nations Food and Agricultural Organization, got a bunch of support there. They were really interested in amplifying this work and looking at other areas of endemic infection and trying to understand the wild bird piece within those areas as well. So through that program, it's probably the first large-scale telemetry data set that was available, almost 700 individual waterfowl marked across 12 different countries and more than 20 different waterfowl species. So I'm going to distill this down quite a bit. We had a lot of paper results that came out with local and international partners, but starting in the top right here of my slide, looking at those bar-headed geese, some of the real basic information we learned was new migratory connections that were not known before. So looking at the tip of that blue arrow is pointing right at Qinghai Lake, and the yellow and orange background there are the migratory Brownian Bridge movement model. So what we saw was that birds had a migratory connection not that far away to Lhasa down at the end of the arrow, which is the capital of Tibet. And up on that plateau, it's where there actually are some people and there are poultry. So previous to this, I think there were only 3 banding records of bar-headed goose from that area, and they linked to India and Bangladesh. So whether in the time that those banding records, between when they were completed and when we did our work, there are changes to the land use and agricultural, climate change system where the geese didn't have to go as far as they would normally, so they're short stopping and wintering in closer areas, or was just not known. But we did identify this new migratory connection. And pairing that with some of the challenge studies that were performed on bar-headed geese, showing an asymptomatic period of around 5 days, about half the birds in the challenge studies, bar-headed geese, died. So some of them still lived, and also there was an asymptomatic period of about 5 days. So what we found from our tracking data is that some of the quickest migrants made that movement within 5 days. So what does this tell us? Okay, maybe these birds, if they can get infected, they survive that long, could potentially move virus, from this area where there's overlap with poultry. And you can see that top figure in the map there with that blue building. Those are captive bar-headed goose farms. It's a really small image, but in the front there you can see some wild bar-headed geese. So, there's certainly on the landscape outside of that Lhasa region, connectivity from the wild to domestic interface there. The bottom right panel, looking across all the tracking data, we could see that there were dispersal movements of birds anywhere from 300 to 1,600 kilometers within 1 to 4 days, whether they're doing, a full migration or they're doing shorter stopovers, is that birds, could move, relatively large distances within, short periods of time, maybe lining up with asymptomatic periods if birds were to get infected. So moving now to the bottom left panel, it's not a really clean, simple story. So depending where we were looking, what species we were looking at, we were trying to look at, investigate the outbreak events in poultry and then the timing and locations of the wild birds. That bottom left gray figure is whooper swans that we marked in Mongolia that migrated down into Korea. And we saw there that the outbreaks were in poultry preceding when the swans had arrived. So, maybe in certain locations it was more so a poultry transmission route than in the wild birds. And then that top figure there, these are some related to the phylogenetic study. So I'll say that this virus information and understanding the genomics is critical for trying to understand. I love Don Hoon's talk, trying to understand, hey, how are these viruses moving by all different mechanisms? Now, one thing that we did learn from some of these, phylogenetic maps that were put out, hypothesizing the movements of H5N1 virus within Asia. There were some mini hypotheses showing or suspecting movement from wild birds to different regions, and we could tell from our ecology work that some of those were not supported. And so the bigger lesson there was that we need to understand the ecology of these wild birds and how they might be involved in actually transmitting the virus. So fast forwarding to today, I'm going to just share a little bit on 3 main things that we have ongoing. So one, we have a U.S.-China collaboration on NSF funding, U.S. NSF and China NSF. The next slide, I'll show you who those partners are. But here's the title, Harnessing Big Data to Understand and Predict Diversity and Transmission of Human and Animal-Infected Avian Influenza Viruses in China. So there's been quite a bit of land use change within different parts of China, and there's a lot more data now available, however, at very disparate geospatial scales, temporal scales. So the point of this grant is to bring in folks from very different backgrounds with different data sets and trying to put it together to make better models to do some of these predictions of understanding the evolution of the viruses and transmission. Point number 2 there, I'm going to talk about one paper. We have work going on within North America investigating high-resolution bird movement data in relation to disease risk factors. And then point 3, at the very end, I'll just mention a partnership we have with the USDA where we are developing transmission risk models, spatial and temporal, trying to understand where the highest areas of risk is at the wild-domestic interface. So on the bottom here are the partners involved in this U.S.-China NSF grant. So University of Oklahoma, our longtime partner, Xiangming Xiao, is the lead on this grant. On the U.S. side, we have Richard Webby from St. Jude's Hospital, USGS here involved, of course, in the wild bird piece. On the China side, we have China CDC, we have the China Agricultural University, and Sun Yat-sen University partners, as well as others. So I want to make a quick plug here. So as part of this effort, we have developed a webinar series, and the point here is just to get, more information out there. Let folks with very diverse expertise share it to a broader community, and just to make the global network stronger. So we have webinars that happen every Tuesday. The first round goes through the end of this calendar year. There's a register link there. We would love if you would like to join us. And we're about to set up the next round of the speakers, so maybe some of you folks would also like to share some of your information there. So moving back to North America, I'm going to talk about this one study where we looked at tracking data of waterfowl species, a blue-winged teal, and relationships with some avian influenza event data. And so the things that we were most interested in looking at here are there linkages between particularly the H5 and H7 outbreak events within North America and bird movements in time and space. And so we were looking at things like that bottom left here, the species distributions, their residency times, that meaning how long was an individual or multiple blue-winged teal present at a given location through time? What was their migration timing? And how does that link up with different environmental and other variables, and also link to disease occurrence probability? I won't go over these equations, but we did use joint hierarchical Bayesian models, and this is to allow us to account for uncertainty within different input variables. And so the first level was to estimate waterfowl residence time and occurrence times. And then to the second level, looking at the probability of an H5 or an H7 event conditioned on the waterfowl residence times. And one of the main points here is that these are spatial temporal models. So with this type of modeling, we're able to account for different spatial dependencies at multiple scales. We've also been able to add down the bottom corner there a clustering parameter that helps to account for more farm to farm versus the more dispersed wild bird movements within our modeling. And so boiling down the results to two main points. One is that the models show that the probability of an H5 or H7 event in poultry greatly increased with the occurrence of these blue-winged teal and their residence time. And there was also higher occurrence of H5, H7 events during the bird's northward spring migration. So that descriptive graph below, what you see is on the x-axis, time. It's basically week of the year, the 52 weeks of the year. And on the left y-axis, we have latitudinal displacement. So the dotted line going across would be zero net movement. So starting there in January, right, any of that solid black line that is going above the horizontal line is a net northward movement. So if you follow that through the spring, birds are moving north, get past the middle of the year, and then towards the fall, they're moving south. And then the gray bars are showing you just descriptive count of the H5H7 events across weeks of the year. And so real briefly here, just showing large-scale zoomed out maps of where the predicted risk is, showing you there at the top right, week 18 being the week that had the overall highest probability of outbreak events, particularly related to the timing of waterfowl. And so about the time that this paper came out back in 2020, there were also three H7 events in the Carolinas. Dong Hoon had just mentioned these in his previous talk. So on March 10th, 2021, there in North Carolina, 2 low-path H7N3 events. And then about a month later, 1 high-path H7N3 on April 9th. And here we're zooming in just to North Carolina, showing you different panels for the weeks from March through May of our models. And here I'm just showing you under that same week with that green circle that our models actually did a pretty good job on predicting these events. And in the top right, you can see that red line is when the April 9th, that H7 high path event occurred. So pretty good linkage in what happened in real life and what the models were predicting. So those models are really meant to show higher risk of introduction from wild birds into the poultry system. Now, there are ways that you could adapt these models to also help possibly if there is an event. And so panel A there is just showing, the 2 low path outbreaks in March. Based on this work, we also found that around 170 kilometers was the threshold for associated transmission, more so farm to farm. And so you could draw, a 170 kilometer buffer around those events. And then in panel B, using the graph down below, could model out the higher, lower risk, depending based on the initial events, and then panel C, adapting the models to both those levels. So basically just allowing some more information where you might be able to target more biosecurity surveillance or being able to follow up for control given an initial event. And so some of the findings of that paper were very interesting. They were H5, H7 events. And knowing from the literature and the bits that we know about blue-winged teal and their migratory connectivity, I'm really interested in trying to break these H5s apart and also look at different species. So particularly for the blue-winged teal, we know from some of the genetics work that they are being found positive with H7 viruses, some linking to events down in Mexico. So some of our next steps are to look at different telemetry data sets, high resolution data, and then also to model out the different H types, H5, H7s, and then maybe grouping some of the other H's as well, and to see what those patterns are, and if we can learn more based on the wild birds here. And finally, just a quick mention here of our partnership with USDA Center for Epidemiology and Animal Health. We're really interested in developing these large-scale spatial temporal models to understand where the higher areas are for risk of introduction of virus into the poultry systems at this interface. And long story short, all these talks that we've heard about, all models are wrong. And we are trying to draw on all these different types of information to help inform our models. So down there in the middle panels there, we have ongoing challenge studies with USDA partners to understand different virus types and different species, to understand the pathogenicity, asymptomatic periods, shedding rates, et cetera. The genetic sequencing is critical, that middle box there, understanding the phylogeny and the relatedness of different gene types to understand the evolution or movement within different events or species. And then we also have some surveillance work on our own, particularly within the Atlantic and Pacific flyways, but also working with the large data sets from USDA and the IRE data sets. Some of that helping to build in weighting factors for the wild bird inputs. That's the top left red figure there, showing us where the wild birds are by different species and spatially and temporally across the year. The right panel on the top there is showing the poultry inputs. So I think that is it, and just please shout out again. Please join our forum if you're able to, and we also have a web page that shows some of the models that we have completed. Thank you.
Thank you, Diann, for an excellent talk. So we're up against our time, and I want to make sure everybody has a chance to get a break. So we will wait until we come back for the facilitated discussion, and we'll take questions on all of the presenters when we get back. So please go stretch your legs a bit, grab some caffeine, and meet us back in about 5 minutes. Thank you. Hi, everybody. Welcome back. So thank you to all of the presenters for some amazing talks today and some very critical information as we move forward. So for the facilitated discussion, we're going to talk about flyway management implications and potential mitigation strategies. We can certainly draw upon the talks today and ask further questions from today's speakers. Plenty of time for that. I also had a couple points to discuss as conversation starters. On the management side, what are our information needs? Are there any barriers to surveillance? Do we have any gaps in our situational awareness? And then on the mitigation side, I was wondering what conditions are conducive to highly pathogenic avian influenza incursions, and we heard a little bit about that today. And what are the ways that we can decrease the impacts of HPAI on our migratory bird population? So what are the steps of prevention that we need to be taking? So I will throw it wide open, and I'll start picking on people if we don't get interaction. And please raise your hand if you would like an open mic.
And as a reminder, you can see the raise hand icon at the bottom right of the participants panel. You will hear a beep tone when you are unmuted, and at that time, please state your name and question.
David.
This may be off topic a little bit, Sam, but I have a question for Dan Walsh.
Go for it.
And I kind of wonder. I think what you're doing with the bird migration is fantastic, but there's a scenario that I don't think these kind of things are going to capture if we just look at migration. And you know, we do a lot of work in Minnesota. Okay, the peak prevalence of influenza in Minnesota actually occurs prior to most of the migration. It's in the resident birds. And my question is, like, how do resident populations fit into that? And number two, does migration really translate to big changes in influenza on the ground?
So yeah, so those are good questions, and obviously we're coming from the perspective of the increased risk that migratory animals would bring, or migratory birds would bring in terms of novel avian influenza viruses. So in this sense, there probably is. I mean, we could definitely see from the eBird data kind of resonant information. So citizen scientists reporting their observations of local birds. But the radar data would not necessarily, you could see some, I guess, flights locally, but yet the intent of this has been focused on those big migratory movements. Small, super, small 20, 30 birds probably are not going to be a signature that we'll be tracking on a radar system. So, so obviously we're coming from a perspective of the risk associated with migration. Now, the risk that resident birds pose is probably a little bit of different question than what we're trying to address. Now, I guess still the assumption is that it would be the migratory waterfowl that would be bringing these novel viruses into the resident populations and then intermixing and spreading that way. So that's definitely maybe a weakness of what we're looking at is how do resident birds affect the risk for poultry producers. We're completely keyed in on the migration side of this at this point.
Can I follow up on that, Sam, a little bit?
Sure. And could you guys turn your cameras on?
Oh, yeah, sure. I've had the pleasure of dealing with the poultry industry in Minnesota during the last outbreak. And the thing that worries me about it, and you are absolutely right, that the poultry industry wants really refined information on when they have to increase biosecurity. And what worries me about some of the risk assessments thing is we will give them a false sense of security. You look at when in the old days in Minnesota when they had free-range turkeys, probably a lot of those outbreaks, maybe most of those outbreaks occurred before migration. And biosecurity needs to be at all times. And I don't think we should bow down to the fact that we need to save money outside these little specific weeks that you say we're at high risk. And I just would kind of emphasize a little care in providing information to them that relaxes biosecurity.
Yeah, so I don't think our intent is to relax biosecurity, and apologize if it came off that way. I mean, I think those biosecurity plans are well established. I think it's more about overcoming biosecurity fatigue, and that is a real issue. Whether or not we think biosecurity should be on all the time or not, it's just people get complacent, right? And so some of this might just be in terms of refreshing people's thinking about biosecurity on the facilities, the diligence they do in terms of monitoring it may be different, those kind of things. But I think the biosecurity plans that the facilities have, and I think some of the USDA folks can actually speak to this better than I can, are generally established well ahead of time. But, I mean, biosecurity fatigue is a real thing, and if there's tools that we can help provide them to overcome that and really kind of refresh their thinking that, hey, yeah, this could be a problem, I don't think that's necessarily a bad thing.
I think we need to remember, though, that, the outbreak in Minnesota started in early March, before migration was really going on. There were not ducks in Minnesota at that time, at least migratory ducks, not many of them. And that gets me to just one other point, and I think it's a little messaging thing. It's a little pet peeve that I have about the way we express things. And I do this myself, so I'm pointing fingers here too. When we say things like the introduction was from wild birds to domestic poultry, we need to insert a human factor in between there. It's a wild bird isolate, but there's a human factor in between that, and we need to make that a clear message.
Yeah, I think that's the point of what we're trying to do here, is acknowledging that it's the human factor, just at the producer level, that it matters. And so obviously, the more information we can provide in terms of risk, and maybe it's not all the risk associated on the landscape, I think that's still useful, that's still important. And as I said in my talk, kind of get away from this vilifying wild birds. I mean, it does still come back to how we do our biosecurity.
May I add to that?
Thank you. Please.
Okay, great. Yeah, I think those are great points, Dave. And I think, so for some of this modeling and trying to understand particularly the long-term migratory movements of birds or the stopping, leaf hopping, frog leaping migration events, right? So maybe that's the movement of some new viruses into the environment. But per your talk, per Chelsea's talk the other day, the environment is really important. And I thought I was running out of time, so I kind of cut my last slide short. But in those transmission risk models that we're trying to do, they are hybrid models. So trying to understand the poultry populations, the wild bird populations in time and space, but adding the environmental component, because that is such a question that, so maybe birds can bring virus into this area. It's in the environment. How did it get into those turkey farms in March? Who knows? Maybe a hunter, maybe somehow somebody was walking around in the environment and the biosecurity wasn't quite clean enough and it got into the poultry farm. So I think you're right. There's different levels for biosecurity and the message needs to be clear. I also think that related to that, to your question on gaps, Sam, we could really use more sampling across the year. And just to understand the different seasonal effects, different waterfowl, maybe in general it's, dabblers as a group, but there are different migratory patterns. I mean, they do all move kind of in a bulk spring migration or fall migration, but I think that is one gap that we can use more information on. Their funding has gone away, as you have mentioned, Dave, so it's only getting harder until probably we get a new event, and then it's going to be thrown on us, and then we're going to struggle per the conversation on the first day on, how to implement it as quickly and effectively on the ground and at a national level. So related to that sampling, we could use more. Sampling can be pretty biased on how we're collecting those samples, rocket netting, swim-in, waterfowl traps versus hunter harvest. So elucidating some of those biases is also helpful in understanding what the results are. Thanks.
Great, thank you, Diann. Russ.
I've got another question for anybody who wants to answer related to gaps. And it really draws on what Diann, you just said, and I think it's really important. We are going to have a major desert of genomic sequences from waterfowl in the next couple of years unless something changes. How is that going to affect everybody's research?
So before we change to that, Dave, I wanted to get one last piece, another crack at the last conversation, and that was, that the potential there for a Shiny app will, I think more than anything else I've seen in the last 3 days, will get the attention of state agencies, agricultural and wildlife. But maybe we're looking a little, not quite broadly enough, as Dave points out. It's true that waterfowl may not be showing up in March, but I'll bet you redwings are. Waterfowl commences, and given the absence of food resources, a little spilled grain around a poultry operation is all you'd need to concentrate birds. So I guess one thought I'd have to, for Dan is, how hard is it to expand the application to, it's a federal jurisdiction, to other species that move back and forth every year. Redwings would be an obvious one. And then the other part, and Dan and I have been chatting back and forth about, this is yet another really good idea that will languish in the absence of sustainable funding. So they can build this Shiny app, but unless somebody houses it, curates it, and makes it, essentially is getting up and doing the care and feeding on a daily basis, it loses its utility almost instantly. So in addition to the surveillance capacities that we've all been talking about and the lack of funding for these things, and now we can talk about genomics, in addition, for some of these applications that will have, I suspect, real currency with poultry producers and wildlife guys and so forth, is how, there's nobody out there with funding that's just sitting around to support these applications that could be very useful. So as we think about developing a recommendation, which might be, maybe that's one thing to come out of this, is a recommendation for sustainable funding. We haven't talked about recommendations from our meeting here, but it might be that a recommendation would be an increasingly pressing need for sustainable funding for a variety of purposes, including field surveillance, genomics, tool applications, and so forth. I guess that's a statement more than a question, but you get the idea.
Thanks, Russ. Dave, did you want to follow up?
No, I think it's a great idea. I mean, that's really the problem, is that we don't have a surveillance network actually set up for the future, and I think we're going to have these huge gaps in data, metadata, and everything else, and even tracing back, even trying to trace back. Okay, we have an outbreak in poultry. Where did it come from? We're not going to have the database to actually look at that. Not with any sort of reliability.
I don't see anybody else's hand up at the moment, so I'm going to pick on Mark Conniff. If you're able to open your speaker line, I wanted to ask you what questions you might have from a Flyway and migratory bird management perspective.
Hey, Sam. Yeah, thanks. Honestly, Sam, I'm, in my particular role, I'm not as attuned to management of disease. I mean, we're more involved in the surveillance, and that's primarily through our Western Canada Cooperative Banding Program. So I was pleased to see in the presentations that a lot of the data that's collected in our banding operations are being used to inform many of the analyses that were presented over the last few days. But really, I think many of the questions that I have pertain more to, at least in terms of my particular role, pertain more to mitigation measures in the field for our crews. How to predict when we should be increasing our mitigation measures, our protective measures, our PPE for our crews in the field, and then also I just lost my second train of thought, but yeah. So I mean, from a field level perspective, I didn't really hear that discussed over the last 3 days. But in terms of the folks that are sampling for AI, with our, and Sam, you've been very helpful in the past in terms of recommendations for our crews and measures for cleaning our equipment and PPE for our banders. But if there is other standard guidance that's being issued to field crews, I'd be very interested in that. So I'm sorry I can't comment too much on Flyway level management of outbreaks.
Great, thank you, Mark. I'd be interested to hear from some of the state agencies what their thoughts are on management and mitigation options. Colin, Julia?
Colin, if you can just go ahead and raise your hand on WebEx, please. All right, Colin, your line is unmuted.
Are you there, Colin?
How's that?
There you are.
Could you ask the question again? I'm sorry. We have two the deer and elk workshop also going on at the same time.
Oh, so this is the avian influenza
Right.
Webinar, and I wanted to know what your thoughts were on potential management or mitigation strategies for HPAI. So we talked about all kinds of really exciting research and results, and we want to know how to apply that in the field and what the barriers to that application might be. And I was wondering from a state perspective what your thoughts might be on management and mitigation options.
Yeah, well, it caught me off guard here. Our biggest thing with several of the states that we work with, and at least on this side of the country, we are forever working in this surveillance mode, and as well as trying to keep in close ties with our Department of Agriculture that find the management of this disease to be, I guess, paramount in their their view, and trying to protect the resource, everything from the backyard poultry producers that of course have little to no biosecurity to the big producers throughout California and up through the the western part of the country here along the coast. They where we end up having some of the outbreaks, and those seem to be some of the biggest challenges is how to integrate the surveillance and management strategies that we see in wild birds into those programs. There's doesn't seem to be a real, I guess, a fluid coordination between wild and the domestic communities and industries.
So One Health may not be working so well in that situation.
No, and I don't think it ever has worked really great. I mean, we haven't seen it. It's just been it's been an issue. We work with USDA and USDI throughout this whole surveillance program from a state perspective. At the same time, the impacts to the wild populations have not been recognized, whereas they are quite substantial, as we saw in 2014-15 to the domestic side of this issue, and the connection between the two is still beyond many states. That how do we utilize wild surveillance to better inform and create better biosecurity across the country.
Thanks, Colin. I appreciate you putting up with me putting you on the spot. I was wondering if Julia Burko or Kristen Mansfield might have anything to add to that.
No.
Chris, are you able to unmute them?
Yeah, but one hit him.
Like Julia, maybe
Yeah, I don't really have anything to add to what Colin said. Yeah, I feel like there's a little bit of a disconnect sometimes, and yeah, I don't know. I don't have anything else really.
Okay. Thank you. Kristen, did you have anything to add? I think that is a no. Just looking for other hands that are up. Pat, Packline.
Hi, thank you. This is really an excellent discussion and wonderful speakers. I wanted to maybe circle back to what Dave Stolnick had commented. I put some stuff in the chat. I just don't know who gets to read that. Regarding the poultry industry, you know, there's always this situation of when you have birds under your control and when you don't have birds under your control. And so in the poultry industry, it's really incumbent upon them, and they have tried to do a better job over the past 25 years for biosecurity. That was one of the things David mentioned about the old days of raising turkeys outdoors in Minnesota. They have generally brought most of those birds indoors unless they're with the National Organic Program, which requires birds to have some outdoor time to meet the requirements of the organic program itself. But for the most part, they brought birds indoors, mostly to break that cycle of transmission between wild birds potentially and poultry. But it is incumbent upon the poultry producers, and not everybody, of course, is equally competent or diligent in keeping their biosecurity. I noted in the chat that the Newcastle disease outbreaks in 2002, 2003 in California and Nevada were actually traced to contaminated farm workers who brought in essence, dirty boots, dirty clothing. They went to fighting cock matches at night, and they basically walked the disease into the farms and moved it between farms in California, and also some birds got moved to Nevada sort of in the middle of the night, I think. But a lot of it is about personnel, and it's educating them. And so anything that we can do on the wildlife side, on the wild bird side, to help them understand transmission routes and infectivity and reservoirs in wild birds so that they can better understand why they need to do biosecurity. You know, it's one thing to demand them to do biosecurity. It's another thing to educate them on why it's important. And so there's lots of different poultry diseases, as you all know, that require all sorts of different biosecurity that needs to be maintained at all times. Fatigue is a human nature, but that's the reason why you have to explain to them why it's so important and where those breaches can happen. So I just wanted to make those few comments. And also for surveillance, they should also be doing the peri-domestics, you know, the pigeons and the little passerines that are constantly around the poultry farms. As I think somebody mentioned, you know, grain on the ground is as an attractant, certainly to rodents and anything else that wants a free meal. So ventilation issues in the poultry houses, peri-domestics, just keeping those poultry farms clean, and also tracking the people that come on and off. So I hope that's helpful. Thanks.
Thank you. Kyle.
Hi, folks. Yeah, Kyle Spraggins, Washington Department of Fish and Wildlife. I guess I'll try to take a crack at this.
Thank you.
The presentations have been very informative, very interesting. I think I'm still struggling to sort of connect the dot back to the sort of overall statement of why HiPath is a concern for wild bird managers in North America. And I'm saying that a little bit tongue in cheek. I understand, except for the fact that there's sort of this assumption underneath this that there is something that waterfowl managers can actually control in this system to influence reducing the risk of spillover. And I'm not so sure that's clear or as straightforward as I think people hope it would be. And I do want to emphasize the sort of the not vilifying the wild bird part. Dr. Stelnick, I think, is right. It's sort of infusing that human aspect part of that link is not the common way that it is stated, and I think that is a communications problem, and that is where I would say my biggest concern starts creeping in. My authority stops at monitoring wild populations, setting regulatory seasons, and responding to those things that happen in public domain that I can actually control. And so in the chat, and I don't know where the chats are going exactly, but there was a question about, are there mitigation factors that could be implemented out on the landscape, like influencing wetlands? Underneath that assumption is that public waterfowl managers actually control most of the wetlands, and that is very far from the truth. The best example of that is something like flooded rice agriculture. Probably very close to 0% of flooded rice is in public domain, meaning the authority for a public agency to influence water out on that landscape is pretty much zero. We do have partnerships that we try to work through and try to balance that, but fundamentally what you're asking, if that was a mitigation strategy, is to reduce the amount of available water on the landscape, which in turn would concentrate the populations further, which I don't think is a desirable outcome. And in terms of the types of disease that we actually respond to, where we know concentrated wild birds is a problem, things like botulism and avian cholera, which when we're in drought cycles like we are in the West, only creep up more, and that's why we're sweating it right now going into this particular winter cycle. There is already conditions on the landscape that are beyond our control, that are going to influence concentration, that we don't have power to sort of undo that concern. And so, but all of that has nothing to do with our ability to influence the spillover. So I really get concerned about the sort of phrasing that it's the waterfowl managers that have the potential to influence the spillover and sort of the ramp up in the domestic side of this equation. And the last thing I'll mention is that the presentation by Dr. Ip yesterday was really interesting, because from infusing the ecological standpoint of what happened in Washington in 2014, it's worth noting the response was based off an aspergillosis die-off, where there happened to be some birds that got submitted for sampling because of the oddity of it. It occurred in December, which Washington sits at a very extreme wintering zone. We dance that freezing line of what would be typical wintering habitat for dabbling ducks. So you already have birds that are in this sort of stressed, pushing the northern boundary of wintering areas state. And the reality is December is about as close to a 0% probability of movement for those waterfowl that you can get in the year. Those birds are not moving beyond that system at that point. They are the birds in that system, meaning the likelihood was it came down far before that, 2-3 months before that, when we weren't actually doing any kind of surveillance, right? So when we're pushed back on our feet and we respond to an event, it then triggers this really easy ability for people to then point back at wild birds. Are wild birds part of the role? Yes. But is that the only part of the role? No. And so here we are, trying to figure out surveillance. And again, there's $0 being put towards this, but all the ask trying to be pushed back on those that are banding waterfowl right now. And that's got to be, we got to do a better job of that. If we're wanting to actually make this a monitoring network, there's got to be better sort of support than sort of hoping that those that are out in the field banding birds are going to be able to make up for the scramble in the end. So I know that was a lot, but appreciate it, and certainly is thought-provoking, if nothing else.
Thank you, Kyle. That was excellent. Craig put a note in the chat box. Craig, would you like to talk to that?
Sure, thanks for unmuting me. It's an important conversation that we're having that has come out of Dan's work and some of the things that David said. There's a critical gap in wildlife health in general, where we don't do implementation science and we don't do human dimensions aspect. I mean, fundamentally, it's been shown over and over in the health research data that the acquisition of new information and facts do very little to change human behavior. We've seen this over and over again with the biosecurity literature and human dimensions, that fatigue issue that Dan brings up is so real and so common, and that we have this huge need to start to understand how do we affect the behaviors that people make. Yes, there are clearly the ecological issues for waterfowl, reserve management, and habitat management, but we're starting to talk about what do farmers do, what do agencies do, how do we make sustainable change. We've now conducted 4 different reviews, and in every review around wildlife health, we found whether it's climate change, the wildlife trade, general research, we do not study the human dimensions of risk management. And until we do this, the conversation of how science will influence decisions is very limited. So I think we have to this goes back to the conversation we had on day 1 about surveillance. Until we understand what people are able to do and willing to do and what information will change those actions, we have a disconnect between our science and risk management.
Thank you, Craig. I believe that Jonathan Sleeman has something he'd like to add to what you just said.
Yeah, thank you, Sam. And actually, Craig has pretty much covered what I was going to mention, and I was actually going to go back to his comments he made on the first day, in that I think what we're lacking is those structured conversations with managers to find out what information do they need, what events would trigger action, and what actions can we actually take. And I'm particularly referring to the protection of wildlife management, of wildlife conservation. And I think until we have those conversations, I think it's going to be very difficult to actually come up with a robust plan. And I'm going to mangle this a little bit. We did something similar. We did a structured decision-making process for white nose syndrome. And this is a very simplistic summary of the results. But basically what came back is managers said, well, we're really not going to take any action until we have a vaccine, until we have some effective tool to manage this disease. And I feel like perhaps we need similar types of conversations around avian influenza. We're definitely very concerned about these new strains, variants of avian influenza and the potential impact on wildlife management, but we're not quite sure what those impacts are right now and what actions can be taken. So I think, to me, that's one thing for us to follow up afterwards is could we look at doing some sort of structured decision-making analysis to start to get at those questions. The other thing that I'd love to see, and this is perhaps more from a scientific perspective, is some sort of risk assessment framework or risk assessment tool. One of the things I struggle with is well, how high risk is this to North American wildlife? And what are the potential consequences? And what's our level of uncertainty? I know, for example, in the public health field, whenever a new variant comes up of an influenza, they sequence it and they can immediately come up with some level of assessment about its potential risk to public health. Can we look at developing a similar sort of risk assessment tool that helps guide decisions, helps us understand the level of risk and the consequences of that risk? Come up with like a simple, traffic light system, red, amber, green in terms of level of risk. Anyway, that's kind of 2 suggestions for something for us to look in the future. Structured decision-making, discussion managers, and some sort of risk assessment tool. Thanks.
Thanks, Jonathan. Andy, you had something to follow on to what these folks have been chatting about the last couple of minutes?
Yeah, I appreciate that, and apologies that I'm backlit here. Maybe I just want to throw out some more fodder for conversation and food for thought. So I guess regarding the comment with regard to managers not having any control or influence on the landscape, I would encourage people to think more broadly about what a manager is. I'm not a manager. I work for a research agency. But I think, private folks managing rice fields, in my mind, they are a manager. They're just managing their own private rice field. Now, I realize that, one's jurisdiction may not be to control water on that person's land, but I think in, say, an outbreak event, this sort of person might be seeking input on what would be sound management. And so anyway, just to maybe encourage people to step outside their individual role and to think maybe broadly about what managers are. Second, I really agree with these points with regard to there's a human component of this that's important. And I think another thing that I encourage people to consider is that managers, wildlife managers, manage not only wildlife, but in many respects, I think they manage people. That is, I think wildlife managers manage people's access to wetlands. So if there's an outbreak in a wetland, we've heard some great talks about persistence of viruses in wetlands. It's not my role to determine if access should be allowed to a wetland where there's an ongoing outbreak, but that's a conversation that maybe should be had. Or if there is access allowed, what sort of disinfection protocols are going to be required as they access those lands? What sort of tracking? Again, too, maybe just thinking about influence and sharing information with other people that manage lands, even if they're not necessarily a public servant. And finally, one last point that I would like to make is I want to really get people to think about the idea that the landscape is changing with these high-path viruses. So, there's been some talk mentioning, how much of an issue is this to wild birds? And I think this is a dynamic situation. This was not an issue for wild birds essentially 25 years ago. And then, starting in the early 2000s, well, this became an issue for wild birds in East Asia. And then in 2014, 2015, well, this started to become an issue for wild birds in North America. So I don't know where this is going. I don't think any of us do. But I would encourage people to think or to understand that this is a dynamic situation, and what we're looking at today might not be what we're looking at 5 years from now. And so I think critically thinking toward ahead about what could happen is useful. Not for getting everybody worked up, but for thinking about as the situation potentially escalates, or maybe not, maybe it de-escalates, that we know we have a better sense of what we might do as we move forward rather than just to be constantly reactive. So I knew I'd throw a lot out there. Thank you all.
Good point. Thank you, Andy. Lisa Shender, you've had your hand up for some time. I'm sorry.
Hi, I just had a comment from my perspective in my previous employment as a state wildlife veterinarian. I'm not with the state of Florida anymore, but when I did work for the Florida Fish and Wildlife Conservation Commission, Florida has a large number of Muscovy duck die-offs every single year. And I think it was one of Dave Stalnick's slides that he showed some data about shedding or infection in Muscovy ducks and how susceptible they are to many different avian influenza viruses. So I guess my comment is, in Florida, there are a lot of duck virus enteritis outbreaks in Muscovy ducks. Are very susceptible to that. At other times of the year, they have botulism events occurring, algal blooms. And so it kind of became a little bit old hat with, oh, it's a DVE die-off. It's, March and April. We know it's DVE. Maybe grab a duck or two from an event and do a quick necropsy and see lesions that are consistent with DVE. And these birds may not necessarily all be submitted for testing for avian influenza. So I'm just kind of wondering about comorbidities and the possibility that these birds might have AI viruses, or that, that could possibly be overlooked. And if there should be increased efforts to look at these feral, non-native Muscovy ducks as sentinels on the landscape. And then along with that, I'm thinking about public messaging from a state agency perspective and how describing duck virus enteritis outbreaks, which are not, a disease that humans need to have a concern about, is a lot less fearful than talking about, avian influenza or bird flu or whatever it might be called. Thanks.
Thanks, Lisa. Those are excellent points, and something that we ran into with the feral chickens in Hawaii many years ago with respect to HPAI surveillance. So there's certainly those feral and peri-domestic species gaps in our surveillance systems. Ross Mason.
Yeah, there we go. So what I was going to suggest, well, there are actually four things. One, again, sort of highlights the stovepipe of what we do. But first off, there is a North American waterfowl management plan, and that plan does not have a wildlife disease section, for example. And so they're talking about management of hunters and human dimensions and wildlife watching and blah, 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 private lands, but there isn't something that actually talks about this area of concern. And so, I think that there may be an opportunity for at least the task force to engage in trying to get something into the North American Waterfowl Management Plan along these lines first. Second, the Association of Fish and Wildlife Agencies will consider a resolution at their September meeting to make the establishment, to make real the National Fish and Wildlife Health Initiative, and to begin working with the federal agencies to create funding one way or another for a national surveillance network. So I'd encourage everybody to think about that. And the third piece that I think is important is someone a little while ago mentioned things that managers have a hard time affecting, but on day one, for example, if you cross the border, 100 miles from where I'm sitting today in Lansing, Michigan, Canadian duck hunters are buying corn in literally truck wagon quantities to put on the landscape to lure ducks for shooting. I mean, you could say it was for sustenance. It's not. It's for shooting. Those things are there all winter long. There are actually some things that managers could do if we aren't just thinking about North America as the United States, but we're thinking about the entire continent and how we could look at that. And then finally, right now there's a lot of effort within the Fish and Wildlife Service and within the states to upregulate private lands management, recognizing that private lands managers, they're not there for the health of waterfowl, they're there to shoot ducks. So we need to think about how we talk about some of these things, whether it's flooded crops like corn in the north or rice in the south, to begin working with stakeholders actually to begin thinking a little bit more deliberately about explaining what we're concerned with and why, because, fundamentally asking people to do things for the benefit of waterfowl is fine, but they're really there to shoot ducks. I'll give you an example. The waterfowl biologists from Louisiana about 5 years ago created a fictitious April Fool's Day thing called the Black Surface Society. And the Black Surface Society was supposed to petition Congress to cover everything north of I-10 with concrete and asphalt so that all the ducks would end up for Louisiana hunters. He had people calling him asking where they could hold a banquet. So when you think about hunters and conservation, they're not necessarily the same thing. I think we do need to engage in human dimensions to think about these things. And it probably would be a great idea if there was a way that state agencies through the Flyways and some of us, the folks on this call, could sit down together and work out some of the opportunities that we have for mutual leverage, both to provide better science, better human dimensions work, and also to begin thinking about what management can be affected on the landscape so that there's this continuity. Again, going back to the notion of a sustained capacity for surveillance. Over. Oh, one last thing for you to mention that, the Flyways are all very concerned right now that because of the ES emphasis within the Fish and Wildlife Service that they are continuing to deregulate the duck banding, duck tracking efforts of the Fish and Wildlife Service to the extent that the Mississippi Flyway and maybe other flyways are sending letters to the service emphasizing the importance of this long-term data set. So, there are aspects within the federal agencies, even for folks that are on this, that they aren't thinking actually about the value of these long-term data sets in terms of these emerging threats. They're actually thinking about whether we actually need to collect the data that we have been collecting for waterfowl, to the extent that states are now concerned with maintaining that capacity. So once again, there's an opportunity maybe to talk to others in the federal agencies about the importance of some of these long-term data sets for the things that we've been talking about. Now over.
Thank you, Russ. Yeah, that's a very important point on the data set. Certainly with West Nile virus, we didn't have a very good denominator in terms of our population numbers to get an idea of how those wild populations were being impacted. Craig, I thought I saw your hand up, but now it's back down. If you did want to make a comment, please do so.
No, I think I just took it down. Nothing more to add. Thanks.
Okay, thank you. Do we have any other questions on the line?
Currently, I do not see any more questions in queue.
Okay. All righty. Well, Andy or Camille, did you have anything you wanted to add, or how would you like to progress?
This is just a suggestion, Sam, so please take your leave. But I think up higher in the chat, there's a number of questions to the speakers from the presentation. If we might have a few minutes to circle back to those before Camille closes us out today.
Okay. So there's a question from Henry. It looks like that's in regards to Dave's talk, whether you could elaborate more on the role of water management on the avian influenza prevention, and whether you could elaborate on the likelihood that we can use serological data to project future avian influenza prevalence in wild birds.
Yes, for us.
Easy one for you, Dave.
Pardon me? Yeah. As far
That's
as
an easy one for you, Dave.
Can you hear me okay, Sam?
Yeah.
Okay. Okay, as far as water management goes, I can't come up with a single way to manage flu through water management that would be tried and true or that I would recommend. We just don't know enough. As far as water management goes and the discussion we've been having, right now I would manage water to actually provide good habitat for waterfowl and not manage influenza. Not even consider managing influenza because we don't know how. As far as the serology goes, we didn't mention, there hasn't been a lot of discussion about serology in this meeting. Where we use serology and where we use serology very effectively is to add another perspective when we're looking at virus detection methods. And to just give you a good example of that, Andy in his talk mentioned emperor geese and how we do not get many isolates from emperor geese. Well, the reason for that probably is that 100% of the adult emperor geese are seropositive for avian influenza and probably seropositive for a lot of different serotypes. They're basically immune. And when we get negative results and see that, we go, okay, we understand it. We know what's going on. And it's a very effective tool for that. We heard some mention of red-winged blackbirds before, and a good approach of going to a species like that. Passerines aren't generally involved in influenza epidemiology too much, but you go into a population like that and you look at virus isolation results. Oh, we got nothing. But then you look at serology, and it's like none of them are seropositive, which shows that they don't even have a history of being positive. Again, you can put it into perspective. As far as using serology to project what may be coming down the pike, we're nowhere. And part of the problem with that is we can measure sort of very effectively measure antibodies to influenza in populations, but there are no good ways to measure subtype specific antibodies. We get estimates. They can't be interpreted. We don't really, it's just, we do not have a way to measure population immunity in wild birds right now, except very grossly, and that's it.
Thanks, Dave. That was a good follow-up on that question. So the next question in the chat box is from Russ Mason to Dan. Dan, you can interpret this one. Fascinating, Dan. So the question is, when?
Yeah, so Russ and I have been kind of communicating back and forth, but we're thinking probably within the year. Now that some of the COVID restrictions on hiring at Cornell have been lifted, we'll have the personnel to kind of finish this, but we should have an application, I would guess, within the year for at least a working prototype.
Great, thank you. All right, Andy, I'm not seeing any other questions there. Do we have any more hands raised or questions on the line?
This time I do not see any questions in the queue.
Thank you. Okay, well, we're close to our time, and I want to make sure we have enough time for Camille to add some closing comments. So I'll just thank all of you for an excellent session of presentations and answers and thoughts from both the panel and the participants. So hopefully this has given everybody a little bit of food for thought, and as Russ was mentioning, we might want to put some of this down on paper. So I'll turn it over to you, Camille.
Sam, thank you so much. I was having a little trouble with the WebEx. Let me see if I can get my video to come on. I apologize. I seem to be having trouble. Can anybody see me, or is the video working?
So Camille, it's at the bottom.
It might be my internet.
It's at the bottom of your screen. Do you see the start video icon?
Oh no, I do see it. It's just not turning on, or at least I don't see that it's turning on for some reason. I'm just having trouble. So my apologies. I'll keep working on this. So basically, I just wanted to close out with some acknowledgments. First, want to thank everyone for your participation today. I want to thank all of our excellent presenters that we've had over the last few days. One of my major thanks goes to Andy Ramey, who really, from my perspective, led the initiating this effort and has been really the lead organizer for this. I think you've done an excellent job, Andy. And then I want to acknowledge my colleagues on the Avian Influenza Interagency Steering Committee who have done an outstanding job not only serving as moderators and presenters, but just facilitating the conversations. And this has been, from my perspective, very rich conversation over the last few days that I know we will continue to discuss as a steering committee to see what the next steps will be. I wanted to let all attending know that we do have a closed session where we're going to be working internally, the steering committee with Flyway Council representatives to see, how we can get the message out and continue this conversation. And I also want to acknowledge and thank from the USDA our excellent facilitators and organizers, Liz Fernandez and her team, who coordinated this on WebEx. We really appreciate their effort and the fact that these recordings will be available. So if you missed out on any of it, please do, once the links are available, we'll disseminate that. And we encourage you to go back and hear some of the great presentations. So that was all I had for closing comments myself. So I just want to pause for a moment. I'll turn to Andy Ramey and others from the steering committee to see if there are any closing thoughts. Over.
Thank you, Camille. Thank you to all the attendees, all the moderators, all the speakers. I really learned a lot these last few days, and this is extremely beneficial to me. I hope you found it beneficial as well, and I'm really looking forward to what may come and follow up to this webinar series. So with that, thank you all for your attendance, and if anyone else has anything to add to close this out, please go ahead and say so now. Thank you.